Good morning, everyone, and thank you for joining us for this discussion about the cutting edge of genetics research. Before we dive in, I'd like to take just a quick poll of the audience, just to show of hands. How many of you, if it were possible today, would want to have a complete readout of your genome and know every possible disease you'd be at risk for the rest of your lives? Okay, a lot of take. A little behavioral economics to start the day. Great. So, well, we'll see what the science has to say about it. Just for some quick introductions, we have Dr. Colleen Campbell, president of the National Society of Genetic Counselors. She also directs genetic counseling operations at the University of Iowa Healthcare System. She works to make sure patients can not only access the information within their DNA, but also understand it and benefit from it. We have Dr. Lon Cardon, CEO of the Jackson Laboratory, a geneticist who's also worked in pharma. His research aims to decode the underlying drivers of disease. Dr. Bernard Kwabi-Addo, a professor at Howard University here in Washington, D.C., a specialist in prostate cancer. He's helped identify epigenetic biomarkers with aggressive tumors and specifically in Black men. And we have Dr. Ben Neal, co-director of the Stanley Center for Psychiatric Research at the Broad Institute. He has led large-scale genetic studies to help better understand ADHD, autism, and a host of other conditions. Welcome.
Thank you.
So my first question will go to each one of you. For decades, DNA research has promised to uncover the causes and the solutions to many diseases, but like any scientific endeavor, the more we learn, the more complicated it becomes to find that answer. So I want to ask you, Colleen, what aspects of the field are you seeing change today? Mm-hmm. And from each of your particular vantage points, what do you see as kind of the main bottlenecks to moving forward?
Yeah, it's a really exciting time because we are seeing a lot of this basic biology and our understanding really start to advance. And as somebody who speaks to patients about these, discoveries and how they could impact their life, we're finally at the stage where we're seeing these genetically informed therapies and treatments come to market and patients being able to access these. So I think it is a very exciting time, like you said. And with the role of the genetic counselors, we're seeing a lot of movement where a lot of providers don't have a really deep understanding of like gene therapy or genetics in general. And so some of the things that, like the newer trends we're seeing is where more genetic testing is happening prenatally so that patients can be eligible when they're born. If they're born with a genetic condition, they can get that gene therapy as soon as they're born and even maybe delivered a little bit preterm so that they can start that therapy sooner. And so the genetic counselors on my team, as well as across the country, are being involved in those conversations earlier to both help the providers understand the genetics and who will benefit or not benefit from these treatments, but also the families, and helping to facilitate that. And they've been a little surprised, I think, where they're more in the let's coordinate the team role because the lack of understanding of genetics from others. I think you asked about obstacles too, was that the other thing? I think one of the themes we heard about a lot this morning in the plenaries was access, and that is definitely the case. So genetic counseling is a relatively new field, and as a result, we still don't have great insurance coverage. Medicare does not yet recognize genetic counselors as a provider type, so Medicare patients have to pay out of pocket for genetic counseling. That's obviously a problem when we're talking about healthcare access, and I think that's one of the main bottlenecks we're seeing. Medicaid coverage is variable across the nation. The National Society of Genetic Counselors just did a benchmark study and looked at that. And then there's a new CPT code, which will be good, but hospitals need this reimbursement so that they can hire genetic counselors and get these treatments and therapies to patients.
Okay. Lon?
Yeah, so I'm interested in the translation of genetic discoveries. And I think in terms of successes, it's good to reflect back that it was only 25 years ago that we knew maybe 8 or 10 genes that cause human disease. We had ApoE4, we had BRCA1 and 2, we had a few of the rare diseases were coming out. And now we're sitting there on whoever can keep track of it, 10,000, 12,000 of these things. So in terms of gene discovery or association discovery for common and rare diseases, This field has knocked it out of the park. I can't imagine something with greater success than that. In terms of challenges, I think we have yet to really unlock that potential and turn that into better treatments or diagnostics or devices or what have you, and that's where we are today. Now, the fact, if I look at this title, Can Genetics Unlock New Interventions? If we're questioning the value of genetics, I have a theory that all of biomedicine goes through the hype cycle. Mm-hmm. And the fact that we're questioning this means that maybe we're at the bottom and coming back up on the translation. Because, I mean, look at everything around. Look at monoclonal antibodies, look at allele-specific oligos, look at AI, look at the microbiome. It all does this. And so, maybe our time for genetics is right now, and hopefully we'll get to talk about it a little bit more. But I believe that one real clear pathway to common diseases is through rare and breaking it down. And I think maybe we'll learn from Bernard a little bit. I think the lessons of cancer have taught us that. Breast cancer 50 years ago when Nixon declared the war on cancer was a disease. And there are 80 histological subtypes of breast cancer today, and you treat them differently. Common chronic diseases, Alzheimer's, type 2, we're going in the same direction. So, thank you.
Yeah, so I think where I see the bottleneck is that there's a narrow window of treatment for many patients with cancer, especially those with aggressive and metastatic form of cancer. Prostate cancer specifically, we don't really have the answer for those individuals whose disease will metastasize compared to those whose disease will be indolent and may require one treatment versus a different type of treatment. So that's a real bottleneck. But at the same time, I'm encouraged with the current technology in really investigating genome-wide differences and all the genetic changes that are being unfolded through next-generation sequencing, through RNA sequencing and protein biomarkers. I think this is really helping us to understand the heterogeneity of tumors that we are dealing with, not only in individuals, but also the intra-tumor heterogeneity, and that's really where the challenge is. But I think understanding that is really helping us to be able to get to the position where combining next-generation sequencing with machine learning is really helping us to analyze, the vast genomic data to really direct us to target therapy. And I think there's a great hope there. We are beginning to appreciate that there are subtypes of cancers, so it's not one size fits all, and that's really what is driving the precision care. So yes, there's a lot of challenges, but I think we're making progress along the right path.
I would agree that genetic discovery has become routine, as Lon indicated. Understanding what genes and variants contribute to any disease, from rare to common, is maybe not fully accomplished yet, but how to accomplish that is extremely clear. We have the technologies, we have the capability, and I think with the sort of emergence of large-scale biobanks with electronic health record information coupled with genetic information, we can actually do this systematically for every human disease. And that's a realizable goal in, say, a 10, 15-year horizon. I think the challenge right now, in terms of unlocking new interventions, is how do we put these genetic discoveries to work? Mm-hmm. And I think there are two parts to that. So one part is, what does the genetic variant do? In what cells does it do its business? How does it change the biological system? What's the perturbation that's introduced? And then the second problem is, how do we actually synthesize across all of these results? Particularly for common disease, things like bipolar disorder or diabetes, we know that it's very polygenic, that there are hundreds to thousands of genetic effects that are all influencing these disorders. And how do we take all of those insights in toto and use them to understand what the biological processes and mechanisms that really drive these illnesses are? And so I think if we can make progress on those two problems, then we will certainly lead to unlocking new interventions.
Right. Well, we touched on this a little bit already, but when we're talking about the pace of DNA research, what happens when it does kind of overtake what's possible further down the line in drug development or the delivery of care? Does anybody?
So you're asking when we make the discoveries, but we're not able to yet get them. And I think one of the bottlenecks there is the policy and regulation, as well as the social acceptance, right? And so regulation's always going to follow behind discovery, but for us to be able to get these drugs approved, get them to the patients, for us to be able to have the patients have that access, I think those are some of the bottlenecks.
Yeah,
I'm sure you guys have ideas too.
So I worked in drug discovery 15 years on the early stage of things, and any of you that also have done things, I find it absolutely astonishing that any medicine we've ever made works. Given the limited knowledge that we have of understanding human biology, it's shocking that these things actually do what they're supposed to do on the tin. I think it's pretty exciting right now, partly to riff off what Ben said, but maybe some of these tools that we've got can help us bring together those pieces of the puzzle in a less linear way. So AI is going to have its great applications, and it's going to be hyped in other applications, but at least can help us think differently about how we put these pieces together to try and do that. And the same with, I work for an organization that has led the world in animal models, in mice models of human disease. They're imperfect. Complement those with the in silico models or with the iPSC model, the human cell-based models. We can put these together and maybe make a little bit better headway into the intervention space.
Yeah, so I think with regards to unlocking new interventions based on genetics, one area that has really suffered is that a lot of the comprehensive data that we have right now, these are all derived from individuals of European ancestry. Mm-hmm. And we are appreciating that, look, we all have differences in how we respond to therapeutic interventions. So we need data from minority population, especially from African American, and also Hispanic and Latinos and other minorities. So in that sense, I think the GWAS idea was to really, of course, look at all the genetic variants. And we know that low-risk alleles, and many of those low-risk alleles actually are predictive of disease occurrence. So that is really helping us to appreciate that, look, there are germline mutations that are actually influencing individual disease susceptibility and response to therapeutic intervention. So we need more of the data from other population, and that's really going to help us to really come up with better therapeutic intervention. But on top of the genomic analysis, there's also the epigenetic changes. And I think gradually we are beginning to get the tools that will help us to begin to study the epigenetic changes. And it's quite interesting that now we are able to detect DNA methylation for cancer in blood. So I think the epigenome actually provides additional promise in terms of providing us markers that we can potentially pursue using non-invasive biospecimen like urine, saliva, blood. And this is really going to help us to monitor disease management and therapeutic response. So I think, it's exciting times, but we still have a lot of work to do.
Yeah. I just want to pick up on a couple of things you said, Bernard, because I completely agree. At the Stanley Center for the last decade, we've been working to sort of internationalize the genetic discovery efforts, and we're working in Central and South America, in Uganda, Ethiopia, South Africa, and Kenya, and then all across Asia as well. And that international spirit is partly driven by the opportunity to discover new genetic variation that happens to be more common in one part of our human family tree versus another part. But it also is what it's going to take to get to the scale necessary to really deliver all of the major genetic insights that are on offer. We have a goal of 250,000 individuals with bipolar disorder sequenced, ideally in the next, say, 5 to 7 years, if all goes according to plan. And the only way to achieve those numbers is to really work in an international setting across all genetic ancestries. The other thing that you flagged that I think is also important is that a lot of the genetic discovery efforts have been focused on diagnoses thus far. And I think that makes sense. I mean, I think understanding why someone gets an illness in the first place is maybe one of the first steps in a precision medicine pipeline. But the questions about treatment response, around course of illness, around severity, around all of those different dimensions that really matter to the patients, right? Someone with bipolar disorder doesn't really care about the diagnosis as much as they care about how often are they going to get a manic episode. And so the more we can move to kind of that level of genetic insight and use that, I mean, particularly for disease-modifying therapies. And I think with Casgevy and BCL11A and sickle cell, we already have a nice example that's coming through the pipeline right now of how you can take a disease-modifying strategy rather than an underlying diagnosis strategy to design a better therapeutic intervention.
Conor, may I go out of turn?
Yeah.
Just because I think for the audience that your two responses to that are really interesting to me, because anybody that was in the plenary and listened to Rob Califf, the FDA commissioner, was asked a question about how to better embrace the entire population. I thought his answer was very interesting because he led to post-approval. So make the drugs and then test them in the broader population, which is sensible from where he said. These two answers were not that. They were better understanding of the disease etiology in the broader population first, before approval. And I think that was an interesting distinction to bring forward.
So what are the main kind of last mile concerns not only when it takes to acquiring genetic samples and information, but also returning that information back to the patients, back to clinicians? Colleen, can you talk about that?
Yeah, definitely. So that last mile, how do we get this to the patients? Like I mentioned, there's issues with genomic literacy amongst healthcare providers. And while there's room for improvement there, they don't have to know everything either. And so one of the things we've done at the University of Iowa, and other hospitals are doing this as well, is embed genetic counselors into the multidisciplinary teams throughout healthcare, not just having them be in a department of genetics, but have them in cardiology, neurology, nephrology. And the way I think about that is, I want my cardiologist to be the world's expert in cardiology, and then have the genetic counselor bring the genetics information to the team. And so I think for the last mile concerns, that's one of the things, making sure that our providers are all empowered to use genomics as part of their practice by having that multidisciplinary team. We also have, like I already mentioned, the insurance issues with making sure patients can access this, and not just our wealthy white Americans accessing it, but all patients have access to this is really critical. And so we do need to pass the legislation to be able to do that. And then also making sure patients are aware and that we're identifying people who will benefit from these. And we've talked a little bit briefly about like the genome sequencing and asking you guys if you would have this. I think we need to see, what do people want? What information do they want from that sequencing? My question when you asked that initial one, my question back to the audience would be, what would you do with that information? So if you had your genome sequenced today, what would you do? What we do know is that a lot of people will have their DNA sequenced or have a genetic test. Let's say it's for breast cancer. They follow that high-risk screening. If they're identified for having a mutation, they follow their high-risk screening protocol for 5 years, and then they start to drop out of the system. Or somebody, even like myself, when I had my daughter, I fell out of the system because I wasn't getting the screening during my pregnancy, and it took me 3 years to get back into the screening that I knew as a geneticist that I needed, but the health system wasn't equipped to be able to monitor and kind of automate that I should be getting that screening too. So I think those last mile kind of roadblocks kind of span all of those things, but making sure patients get it, they are able to use that information to actually prevent disease. Hereditary cancer is preventable. Hereditary heart disease is preventable. But we need patients to be able to access not only the testing, but the downstream MRIs, mammograms, prophylactic surgeries that come after you have a genetic diagnosis as well.
Yeah, if I can add to that, I think health literacy is a big one. And I think we're at the stage that we need to be able to communicate cancer at a basic level, to the stakeholder. Because I'm from West African descent, and I typically work with a lot of people in the DMV area. And in my population, I think cancer is a death sentence. So people don't even want to talk about it. It's like, oh, this is my fate in life, so I'm going to live with it. But when you get a chance to actually, in a, let's say, a church setting or community setting, and you begin to demystify that whole thing, they become more receptive to really understand that, oh, there's a lot of things that I can do to modify my risks. And you begin to tell them the importance of adhering to age-appropriate screening. So health literacy is a very important area that I think we need to deal with, especially in the minority population. Yeah.
I think there's we have to bring up the teaching side of this, and not just for the return of results to patients, but to physicians, and we often talk about that. And I'll pause for a second, but ask yourself in that, how many clinical trials are ongoing for cancer right now? Just ask yourself for a second, because I asked myself this a couple of months ago, and somebody looked it up. 20,000. What physician could know everything that's going on? It's not knowable by a single person. So we have a training component to do it, but there's nobody that can. And we started something that I'm really proud of, but I think it should go. Maine is very rural. There is no traditional medical school. The hospitals are a practice, but people get cancer just to say it's an aging population, they get the same thing. So we put together something that was sort of crowdsourcing results. So any patient gets diagnosed with cancer anywhere in Maine, they get sequenced. It comes together, it gets reviewed by a tumor board. So you crowdsource all the people together, find out that there's a trial in Iowa, point the person in that direction. Survival curves just came out for the first time, saving lives. It's really, and no technology, no new genetics, it's just changing healthcare practice. And I think those type of things are really, really exciting to see forward.
Yeah, I am curious, when do you all expect we will see more health outcomes data tracking the impacts of genomic testing? If it's going to be prenatal, if it's going to be as an adult, or even direct-to-consumer testing. I don't know. What do you think?
I would say sooner than I would have originally expected. And part of that is there's been an increasing number of population genomic screening programs happening at state levels, happening at academic medical centers, where they're looking to sequence, I think Alabama was the latest to announce this with their Catalyst program, but they're trying to sequence large portions of the population and return those results to patients and then see the outcomes. And so like Health Partners in Minnesota is doing this and working with, the payers on what are these outcomes. I think as those population health programs take off and we do see more sequencing and, like Ben said, integrated with that electronic health record data, we're going to be able to follow the outcomes better. And especially if those programs collaborate with us, we'll be able to see more outcomes probably starting as soon as I'd say 5 years, if not sooner. I don't know what you guys think.
Then I want to riff on that.
Yeah, I think we're already there in cancer, right?
Yeah.
You look at the survival rate in cancer, that is driven by sequencing more than anything else, I think it's fair to say. And I guess I would also comment that in cancer, we're almost in the upside down world where the clinical practice is ahead of the science curve in terms of the adoption of sequencing technology. And that doesn't, there are a lot of things that contribute to that, but I think it's an interesting place where there's, in a sense, more data in the clinical system than there is in the scientific system. Right. And I think that's the other way around for common complex disease, where we have a lot more information on the research side than is being used in kind of clinical practice. And there are thorny issues to work through with respect to how we think about polygenic scores and kind of summarizing the risk that your genome confers to you. But not just summarizing it, but also, in a sense, parsing it, because polygenicity is a mathematical construct. It's not actually a biological phenomenon. It represents average, increase or decrease in risk summed over the entire genome. But it's pointing at individual biological effects, distinct physiological processes. And I think part of what we need to kind of get better at is how we use those kinds of insights to then try and improve the way that we deploy medication or engage in preventative care. I think all of those things are on the table, but it's just going to be a bit of fits and starts. The last thing I'll say is that when I do see the kind of small-scale healthcare outcome improvement question from using genetic information, it's actually going via behavioral modification, because the doc sits the patient down and says, well, we've looked at your genome, and it says you need to eat right and exercise. And the patients respond to that better than if the doc just says you need to eat right and exercise. And I just want to make sure that everyone here is eating right and exercising, because that's still the standard of care.
Yeah.
So, there are a lot of angles on this, but I think using information and using personal information does resonate in an important way, and that can be very powerful in clinical practice.
Can I comment on that? I would agree with cancer, and especially that the clinical's outpacing. We see this with the guidelines, right? NCCN is coming out with guidelines constantly, and it's really hard for providers, especially if you're in a community hospital, to keep up with the rapid advance of guidelines. And I know I'd be interested, Bernard, your thoughts on this, but for men with metastatic prostate cancer, there's a guideline saying you should have genetic testing. Vast majority of men that are not getting that testing, but there's a lot of, you can have the guideline, but how do you implement the guideline? How do you help the providers be aware and get access? I don't know if you see that also with metastatic prostate.
So I'm a basic scientist, so I'm a clinician, but I think I'll just chime in on that. So I think, yes, men are black men, being encouraged to have their genetic profiling done in the metastatic stage. But here we don't really have, what is the reference? The TCGA clearly is skewed to individuals from European ancestry. So I think we still need to get to that stage where we get more data from men of African ancestry. Now, a lot of black men self-identified actually in the US are 20% Caucasians. So that actually also skews some of the data that is coming out, the polygenic risk score and other modalities. So it's great, but we still have a lot of cleaning up to do to really be able to get to the risk alleles that are really predictive of aggressive cancer, for instance, in one population versus another. So we still have quite a bit of work to do, I think.
Great. Ben, could you talk a little bit more about maybe the specific challenges seen in mental health research and approaching that from a genetic view?
Yeah. So I think, mental health is a huge public health problem. It is the single largest source of disability worldwide, and it's really hard to work on. It's like as hard as it gets. In cancer, you get to take a biopsy, and then you can study the biopsy as your kind of model system. It's very hard to sort of take someone's brain and study that as a model system. It doesn't really work that way. And where we are in neuroscience is nowhere close to where we are in something like immunology. And genetic discoveries are fantastic starting points for trying to understand how disease operates. But they work best when you have a kind of rich biological ferment to land on. So a concrete example of this is a gene called TYK2, a coding variant discovered in the first wave of GWAS about, say, 10, 12 years ago. And it was just, maybe 15. And it was discovered to confer risk to psoriasis, rheumatoid arthritis, type 1 diabetes, like you name it. And now we have an approved medication from BMS targeting TYK2 for psoriasis as a TYK2 inhibitor. And the reason that moved very quickly, like 15 years from genetic discovery to therapy, is a pretty good timeline in drug discovery. I'm sure Lon would agree with that, is because we understood what TYK2 was doing, who it was interacting with, what cell type it was actually doing its business in, why it might actually lead to excess inflammatory response that might drive autoimmune disease. And so one of the big struggles in mental health is that we don't have the same kind of rich biological knowledge about how the brain works. At a cellular level, we're getting better. So the introduction of single cell technologies is teaching us what genes operate in which different cell types. But I wouldn't say that we're really anywhere on the physiology of the brain, how we think about the kind of electrophysiology of the brain, how the circuitry is wired, how it reconfigures itself, how neurons talk to other cells in the brain, how they work together to create behavior, how they work together to create sleep, how they work together to create mood. These things must exist as quantifiable phenomena, but how we actually understand that is what's going to be required for making an improvement. That being said, strong-acting rare variants in particular may provide a shortcut, and they may provide a shortcut in two ways. The first is that if you identify a subset of people with a strong-acting mutation, it's like you found a new rare disease. So it's like fracturing this common disease, as Lon was saying earlier in the panel, into more coherent subsets. And that might not only jumpstart biological investigation, right? So rich clinical workup, try and see if there are any obvious biomarkers, try and identify something that the pharmaceutical industry can actually leverage as an intermediate endpoint before the final endpoint of actually improving the course of illness. That's very promising. And then over and above that, if we can convince ourselves that those mutations have quantifiable physiological effects in, say, model systems, be they mouse or cells, and we can reverse them by editing that mutation out, that may give us a leg up on at least addressing that subset of individuals that carry those kinds of mutations as a way to sort of get to the 20,000 clinical trials for bipolar disorder that we see for cancer today.
Lon, could you talk about the model work that you're doing at JAX?
In terms of the rare.
Yeah.
So that is a terrific example with bipolar right there, because I would go out on a limb and say, despite having said genetics has had all these terrific successes and GWAS has annotated the genome for us, from a drug discovery perspective, I don't know what to do with that information. I don't know how to turn that into something that leads to new interventions, except the way Ben just said. A reductionist approach where we take what is bipolar, and there's probably 100 different paths to get to the same manifestation clinically at the end. If we can start breaking those down and find the etiological components, then we can study them in other organisms, in models of disease, study the genetics, and that's why I went to the Jackson lab, because I think the future of drug discovery from a genetics perspective of common diseases has to be the way Ben just described it, through rare. That's how we're going to make the inroads. And if we get it right, it's a real rosy world, actually, because if you find those patients for whom the treatment is relevant, the response rate will increase, the trials will decrease in size, and they'll be faster because the effect size is bigger. If the world works right, that translates into drug pricing and access to rarer subcomponents of the population, and everybody wins. So I think that's exactly, for all the diseases, actually. It's not going to work for everything, of course, but I would posit it will work across a broad swath.
How do you see that rate accelerating even further with new in silico tools that are being developed today?
Yeah, I do think, I think there's a magic, I hope, to be unlocked between human cell models and differentiated into the right tissue, driven by better quantitative tools and linked with informed usage of whole organisms, whether they're mice or zebrafish or whatever is relevant for that disease. Putting those together has to be better than any one of those individually, and it has to be something that we can use. We can modify these things. I mean, I'm sorry, bless you. Yeah. I'm often reminded, because I've been around long enough, it was not that long ago. Think of Huntington's disease, the second monogenic clone, 1992. The way they found Huntington's disease was to find people observationally that happened to be harboring a mutation. And they drew out the genealogy, the pedigree of those people that would really span this entire room, 18,000 people in Venezuela. It took years and years and years to do it. And now we can go in with editing in a dish, and an undergraduate can do it in a day. And so that pace has to impact exactly the type of disease reduction that we're talking about here.
I think there's a lot of promise on in silico methods. But it is also, in a sense, research, right? So one of the funny things about research is that we don't know when we are going to hit the plateau of utility. And there's some chatter now that maybe the very, very large language models are hitting some kind of plateau, that we keep adding parameters and we keep running more and more compute against those things, and we sort of are maybe maxing out and maybe even going the other way because enough of the data that it's being trained on has been coming from the models rather than coming from humans, and that's starting to make the model, in a sense, eat its own tail. And that does not feel like the way that we're going to get to a solution. I just want to call out one very specific area that I think is very meaningful for the problems that we're working on, and that's really coding variation and really coding variation that changes the amino acid sequence of the protein. So it's changing, it's changing potentially the structure of the protein, potentially the function of the protein. And I want to highlight that our annotation of these mutations is still incredibly limited. That we know that there are probably 2 to 3 times as many of these mutations that really matter for disease as there are things that knock out one of the 2 copies of a gene. And we still cannot predict them right now. The only way that we figure them out currently is we get enough people together with the same kind of illness that all have exactly the same kind of mutation. I'm sure Colleen can speak to that.
Yeah.
Or we look in families and we see that this is a newly arising change that neither of the parents have, but just the kid has. And that newly arising mutation is, in a sense, a functional filter. It tells us that this mutation is more likely to matter. It's not a guarantee. There's a baseline rate of that, just like there is of pretty much everything. But using that, we can actually identify genes where the primary mechanism of risk is operating through these missense mutations. Now, I kind of walked you through that example primarily because that's where I see computation having a very near-term impact, that the structural analysis of proteins is getting pretty good. That information is now feeding back into the genetic discovery cohorts to try and enrich signal to figure out what genes and mutations matter. And it's starting to work, but I don't think we're all the way there. And there are a whole bunch of other things that we want to predict that we aren't predicting now, like what are the biologically active parts of a protein. That's a different question to what shape it makes. And that kind of question, I think, will also get consumed by AI to some degree. But at the end of the day, you still got to show it at the bench, right? Drug development is not a probabilistic outcome. You're putting something into a person, trying to make them better. And that comes with risks and potential upside. And so, it's important to be clear about where these models work, where their limitations are, and how we can use them judiciously to try and accelerate the drug development pipeline.
Yeah.
Well, I think it's interesting we're talking about, you mentioned kind of plateauing a little bit on the data, but we've also been talking a lot about underrepresented communities in data sets. Do you see that extending the runway when it comes to those kind of computations? Or is this, as the cost of sequencing has gone down and become more accessible to laboratories, are we also seeing it become more accessible to different populations?
Yeah, so I think, it's changing because clearly people are more aware that we need to include different populations in these studies. We need new models to really study different populations. So I'm talking about a lot of the commercially available, let's say, cell lines that we routinely use for functional studies. Most of these are from Caucasians. So we need other groups. But I'm also excited that now it's getting easier to do patient-derived organoids models. So you're able to get different models from different populations. And I think all that is really going to help us to really understand how, let's say, a driver mutation is really influencing the biology in one particular model versus another. So those are very interesting discoveries that are coming up, and I think that's going to help us to really understand the disparities that we see with regards to a lot of these diseases.
Yeah.
I would just add the more kind of just basic supply chain, the more people that are having the sequencing is driving down the cost, which is allowing better access for more patients, but also it's enabling these larger research as well as clinical diagnostics programs to develop where they can sequence somebody's genome for $200 and return those results. It's probably going to cost you a little more than that to do the whole package, but that is becoming a reality where we can get these statewide programs up and going. So,
And Colleen's going back to sequencing, and I think we're talking here about rare diseases, and just to baseline everyone, there's people argue about it, but somewhere right between 7 and 10,000 known today. So, and they're really interesting because if you look at the prevalence distribution of those, how many patients are in each of those, about the top 80 or 100, you can carve it however you like, probably have numbers that would be big enough to attract an investment from a drug company to make money because they could get a return on their investment to make the drugs. But that means 9,900 don't. And the tail goes off to where there's 20 patients, 30 patients, 50 patients, and you can speak to this really, really well. But one, I don't know if this is fact, but in my observation, every time, so you, for whatever reason, you get a drug out there and then you start sequencing, the prevalences go up because you start finding more patients with those relevant mutations for that drug. And so actually it gets more accessible in those rarer domains, and it's super exciting to see. So I think that's going to be one of the effects of, it seems to me, of sequencing.
Yeah, we definitely, with sequencing, have seen what we thought was disease A and disease B. And when we're able to sequence the genes, we actually realize it's a spectrum. And you have patients, probably with modifiers that you guys are going to discover, that change how they present in the clinic, but they're all caused by the same underlying gene not working the way it's supposed to. I'm also thinking about like the AI, and this patient just popped into my head where I was once giving back sequencing results. It was the second child in the world discovered with this. So it goes back to Ben's point about like, you identify one person and you have one person, but then how do you know that that's actually causing the disease? You need more people with the same underlying genetic changes. So I'm telling this mom that we found what we believe is the cause of her daughter's disease. She's the second in the world. And the whole time, the whole appointment, she's on her phone. That's not unusual for patients who have been on a diagnostic odyssey. Families know today's the day where you're going to get the answer. So we often have, other people calling or coming to the appointment. So I assumed that's what was happening. And so we got to the part of the appointment where I said, so have you talked about who you're going to talk about in your family? Who are you going to tell these results to? What are you going to do with the results? And she said, oh, I've been taking notes this whole time. My 2-year-old daughter has 2 million Facebook followers, and I have been posting on Facebook. And this was this mom's attempt to find other people like her daughter. And I'm thinking about AI tools, and this is almost 8 years ago, and I'm thinking, wow, today it'd be so much easier with some of these tools for her to be able to find people like her daughter so that they could get the attention of the drug companies to be able to find, a treatment for this rare form of epilepsy that she had. And there's just different ways that patients are trying to find those other patients, find the drugs, and it starts with the rare, where we can also understand the biology. It's very cool.
Yeah. Right. I want to kind of follow that up with one of the questions we have submitted from the audience. I believe there's a QR code, or you can use the app. Ben, many mental health conditions are diagnosed subjectively. Genetics kind of promises to make that more objective. Do you really see it enabling a precision medicine approach?
I mean, I think so. I think the class of strong-acting rare mutations that change the chance that you get bipolar disorder from, say, 1 in 100 to 1 in 10 or 1 in 5, that's a pretty big knock to the biological system. That has a really big impact, and it, it's not a guarantee that it should point precisely to what the intervention should be, but I think for a subset of them, that's a realizable goal. I think the other thing that I just sort of want to flag is that the diagnostic system that we have today for psychiatry is sort of an evolving consensus of, frankly, largely old white men. If I'm going to be completely explicit about what the APA body looks like, I mean, it's changing a bit now, but that's more or less where it came from. But a lot of the illnesses that we're talking about have thousands of years of sort of documented history. Like, you go read your Galen, and you can get at things that look remarkably like anorexia or remarkably like schizophrenia. So there is a common thread throughout our own recorded history that points to some notion of some of these illnesses. The genetics, however, says that the genetic risk factors that we find it is as likely as not that they confer risk across multiple different diagnostic categories. So we see genes that are implicated for autism and bipolar disorder and schizophrenia. We do see some that are more specific to, say, schizophrenia and bipolar disorder and don't seem to show so much in the neurodevelopmental disorders or the autism kind of landscape. And I just kind of want to pick up on that sort of schizophrenia bipolar piece for a bit because, so there's this gene that we've discovered called ACAP11, loss of function mutations, clearly associated with bipolar disorder and is also sort of cropping up in the schizophrenia analysis. Now, we don't really know very much about what ACAP11 does other than it seems to bind with another protein called GSK3 beta, which is the thought target of lithium therapy. And I raise this because mood stabilizers are generally not given to people with schizophrenia. You normally try antipsychotics to start with. But given this kind of genetic insight, the psychiatrists see that and they immediately ask the question of, well, maybe we should be thinking about trying lithium in some of these individuals that have psychosis, so they lose touch with reality, but they also have some mood disruption. And I think that's the other kind of part of this is that there are many prototypical cases out there, people that fit all the criteria, that tick all the boxes, that look exactly like your kind of case of schizophrenia or your case of bipolar. That's super clear. But then there's a whole bunch of people that don't fit neatly into these categories. And the clinical system needs diagnoses to govern questions of reimbursement, of expectations of course, of treatment. It is a decision-making entity, the healthcare system. It has to make decisions about what interventions, what care is going to look like. That's not the same as a biologist's view, where we allow ourselves more of a kind of spectrum, that we don't need these crisp, discrete lines because we don't have to make the decisions to inform on care. We just try and understand that which is in front of us and embrace the reality that we perceive to the best of our ability. And so part of the issue here is diagnoses are necessary because that's the way our healthcare system works. But all of the psychiatrists that I talk to recognize that there are clear limitations in where we are today and that they are doing the best that they can to treat the person in front of them. And so the more nuance we can bring to the underlying root biology, the more clarity we can bring to likely treatment options for those individuals that carry these mutations and maybe develop new interventions through that, and then bring that to the clinicians in a way that rather than adds complication and overwhelms them in data and makes it hard for them to do their work, but actually provides more rapid, ideally computationally informed recommendations that can help improve the management and treatment of these illnesses. That's the future for precision psychiatry, I perceive. And I think we're on that road, but psychiatry's hard. It's kind of always been really hard.
Can I just add to that? I agree with everything Ben said. And psychiatry, genetic counseling is one of the newer emerging fields. And what, back to Ben's earlier PSA about eat well and take care of yourself, sleep is included in that. And what the genetic counselors in this space have been finding is that the diagnosis can also be important because regardless of what disease it is, if you sit down and ask a patient what caused your disease, they will have a theory. Most of the time, they're going to have a theory. It's because I did X, Y, or Z. That's why I got cancer. That's why I got schizophrenia. And when you can say that there is a genetic component and explain it's not all genetics, it's not all environment, and help people work through that, even if you don't identify the genetic component, then they may be more likely to follow Ben's PSA advice about eating and sleeping well and taking care of themselves, which is really important for mental health. But if they think it's genetic and there's nothing I can do about it, they may not follow that guidance. Whereas if they understand these gene-environment interactions, and that's where although there's not as many diagnostic tests in this space, a lot of the genetic counseling is around that and helping people understand it's not all or the other, but it's this combination.
No, I completely agree. And the jam jar analogy from Janine Austin is absolutely exquisite. It really lays out that you have some ability to engage in health-seeking behavior that can actually improve outcomes. And if you are trapped thinking that it's purely genetic, that kind of can make you feel powerless. Yeah. But If you also think that it's purely environmental, then you maybe feel guilt and shame about all of the things that you've done. And so trying to find that middle ground between these two poles is really important and can be really empowering for people struggling with these illnesses.
Just to extend on one point, if I may, that you raised there, Ben, that it is becoming commonplace. Geneticists would call it pleiotropy, but it's genes that are identified for some disease that have an impact somewhere else. So from a drug discovery side, if you ask yourself, for any gene, for any drug target, what is the biggest predictor of success in making a drug out of that? Well, the biggest predictor of success in drug discovery is that someone already did it with that target. So you come late because it's already been disproved. The second biggest predictor of success is genetics. Is there genetic evidence for that target? And so the pharmaceutical industry's heard this, and so you go look at pipelines, and there's often various people will write about what proportion of the pipeline today has genetic evidence, and it's getting higher and higher and higher. The funny part about that is a big proportion of those drug targets that have genetic evidence are being studied in clinical trials for a trait that is not the one that the genetics points them to. So I think when you get to something as complicated as bipolar and mental health, you got to listen to the data. And I don't think that we do as well as we really could.
If I can also add to that, so through epigenetics, we see that genes like ACE, which is the angiotensin converting enzyme, of course, typically we study this in renin-angiotensin or heart disease, right? But we see that individuals who are smokers, when you profile the epigenome in prostate cancer, this is one of the genes that is altered. So this begins to tell us that this ACE is actually modulating maybe the androgen receptor signaling pathway. But it could also play a role in the high incidence or high risk of prostate cancer on patients who are taking antihypertensive medication. So it's quite interesting how one gene, which obviously has absolutely nothing to do with the prostate pathway, is actually using the epigenetic mechanism to modulate therapeutic response.
Interesting. So just going back to the bottleneck question we had at the top, do you feel like the question of moving forward is we don't have the proper tools today, or is it that we're not asking the right questions or asking the questions in the right way? What do you think?
Everybody will have their own answer to that. I have yours will be really interesting to hear. I'll give you mine really briefly. I think technologically we have exquisite tools in biology. I think Ben raised the point that we need to understand the functions better, and there's a lot of work to do there. But we have a really good look at the human genome. And we link that really exquisite microscope, if you will, to clinical phenotypes that we've been looking at for 100 years. So it's the phenotype side that is the clumsy part for us right now, far more, in my view, than is the technology and the genetic side of things. Others may have different perspectives.
I think there's room for improvement on the technology side, too. I mean, so, a bunch of the genetic discoveries for schizophrenia and bipolar disorder point to gene regulation. So proteins that bind the genome, bind DNA, or modify splicing of RNA. These are complicated, interesting biological pathways that people have been studying for many decades. And a sort of consistent question that comes up in prosecuting these kinds of discoveries in a cellular model is, well, the epigenomic landscape of every cell type has some difference, right? So it's structured differently, and how good we are at recapitulating the cells in our body when we try and make them in a dish is still a work in progress, I'd say. I think we need an improvement in how to think about making sure that these cellular models are recapitulating the biological processes and phenomena that we need to understand to translate the genetic insights. So I think there's definitely room for improvement there. It's a lot of activity in that space. It's very, very exciting. And then the other thing that is very challenging, certainly when we come to human-specific things, and particularly when we talk about mental illness, is how to go beyond cell autonomous function. So immunology is great. You got a whole bunch of cells that are circulating in your bloodstream that are all doing their business, basically independent of their environment. You can study, you can go out, isolate those cell types, understand their functions, and it will largely port to the human system, modulo some questions about resident immune cells. To make neurons behave truly like neurons in a dish, we are not there yet. They're not firing in a synchronous way in the way that you would expect them to based on when we take like a slice of brain from a model organism and put it into a dish. That looks very different than what we have the ability to manufacture right now. So I really would put some emphasis on technological development to make these tools more truthful to the human system. But I'm pretty upbeat about that future.
So I think from a basic science perspective, the tumor heterogeneity poses such a huge challenge. And I think we need to be like 2 steps ahead of the tumor. So we need to be able to come up with novel markers that will, modulate treatment, disease pathway. So as you are treating the patient, because the treatment itself induces additional genomic changes. So we need to be able to monitor that. We need to be able to know the individual's family history, their diet, lifestyle factors, and all that we need to incorporate it into some new algorithms to be able to help us to be on top of treating patients with cancer.
Yeah. So I think as the clinician up here, from the bottleneck perspective, I definitely think we need to help providers be able to know when to order genetic testing and for who, and who will benefit from it. And so I think having, that's where AI can come in, and having tools built into the electronic health records will be really helpful, so that as we develop more and more guidelines, as these guys figure out the basic biology of these diseases and it's getting translated, we can make sure it's getting to those patients, because it's going to be the 20,000 trials is a real issue. And then I think we also need to make sure we keep working on the education, both of providers as well as patients, but that education needs to be tailored really to them. A patient wants to know, how does this impact me now? Last week at the American Society of Human Genetics meeting, Wendy Chung was saying, news you can use. Like, what do these results mean? How are you going to use it for your health so that you make those interventions? And then I go back to the access issue. We need to make sure that patients have access, and researchers, actually. I was thinking back to during my PhD listening to you guys, and like when I would take the family histories as a genetic counselor and then go back to the lab and look at the results, I was making connections, like you see families where there's multiple people on blood pressure medication at 18, you see a gene mutation in an aquaporin or a water channel gene, you start to make these connections between, okay, there's some fluid regulation issue going on. But I think having patients and physicians have access to genetic counselors on their team to really help bring that genetic information so that they don't have to be aware of the 20,000 trials, but the genetic counselors can help with that, I think is really key. And to make that a reality, we need to pass the Access to Genetic Counselor Services Act and other legislation to make sure that hospitals are able to hire genetic counselors.
Right. I know we only have a couple of minutes left, and I promised to get everybody out of here in time for lunch. But I want to go to one more submitted question. It's how can the average person or patient take the first step toward understanding the context of their genome in relation to potential or future diseases that may or may not be diagnosed at any point in time? And how can they kind of contribute to the research?
Yeah. I'll start off on this one. So I think the key there, and what some of the issues are with direct-to-consumer testing, is you really need to take your genome by itself is not the answer to all of your questions, right? It has to be put in the context of your medical information, your family history. All of this information has to be looked at together. So the first step a patient could take is to meet with your physician, meet with a genetic counselor who can then help you put that genetic information into the context of your overall health. I think that's the number one thing you can do. Trying to do it on your own, there's a lot of companies out there that will say they can help you do it on your own, but it's really difficult to do. We might be able to say you have a genetic mutation. It doesn't mean we can predict the severity, the age of onset. You really need to look at that. But when we start looking at all those pieces together, we can say, okay, you're more likely to probably get this in your 40s based on what we know about the biology of the disease and your family history. I think the other piece of it was how to get involved with research. Researchers are very excited for you to get involved with research. There's a lot of protocols out there. There's large ones like the All of Us program through NIH as well as others. And so reach out to researchers on an individual basis. If they don't know the study, they'll know somebody else, as well as joining these larger scale projects.
I have a quick question for you on
Yeah.
that same thing. So that question to me reflects a little bit of the journey that many rare disease families
Yeah.
hear. When they get that news that, enjoy the time you've got because it's limited and those are the next steps, then they become the most informed person on the planet.
Yes.
And they go, your story about the person Facebooking and so on. But when I talk to those folks, what they often, almost always tell me is, we really needed somebody to help us navigate. Because we didn't really know. We didn't know the clinical community. We didn't know who to talk to in genetics. We can raise a bunch of money, but we might squander it because we're not spending it wisely because we don't know how to do that. Do you provide that sort of guidance for them all the way through?
Genetic counselors, yes. So genetic counselors do try to provide that guidance. So you might be diagnosed with something, you're aware of some of your symptoms. Based on that gene and what these guys have been talking about, we may also realize that you are at risk for other health conditions. And so we can make those referrals, help people navigate that, help them identify the clinical trials, help their providers understand the results, because often if you're like the second in the world, your doctor may not know what it is. And so we do try to provide some of that navigation, but I do think there's room for that genetic navigator role to continue to grow, and we'll see that as the workforce grows and we have better access. But that's definitely the case.
Well, thank you all very much for joining us, and thank you for coming. Thank you.